Hello students. So you can just see here a new topic, transport across plasma membrane. We are going to discuss this today. Uh, in the last video, I just have told about the plasma membrane. So in that video only, I just have used a term selectively permeable or the semi-permeable. So what does it mean and why is it necessary? है ना? It means that it will allow movement of only certain substance. है ना? Not all substances will be allowed to be easily passed through the plasma membrane. है ना? So if the membrane suppose lose this selectivity and it randomly start allowing all the substances that are coming across it. So what will happen? The cell will no longer will be able to maintain homeostasis, or you can say it will not be able to sustain itself. है ना? The cell will be destroyed. Cell will be destroyed. So that's the reason it is very, very, very essential that the transport across plasma membranes should be regulated. You know, so you can just see here in the previous class we have discussed that the plasma membrane is a bilayer of phospholipid. So you can just see here the hydrophobic like that of the oxygen, CO2, nitrogen, benzene, all these will be easily passed through the membrane. है ना? In this green, you can just see the difference in the thickness of these arrows. This is to tell you that these can easily pass. The one that can easily pass is made with the thick, shown with the thick arrow. Then the smaller uncharged polar molecule like that of the water, urea, glycerol, they will also be allowed to pass through the plasma membrane, but not as easy as that of the hydrophobic molecule. So you can just see a slight thinner arrow. Then the large uncharged polar molecule like that of the glucose, sucrose, they will feel some difficulty in going through such a plasma membrane. Then the ions will not at all will be allowed. है ना? So that's the reason we have the for the glucose we have glut transporters are there. Na? So we will see. So first of all, we are going to just see the passive transport. So here in the passive transport, transportation occurs along concentration gradient. No energy is being involved. है ना? More accurate, you can just say along the electrochemical gradient. For the charged molecule, instead of concentration gradient, the best word will be electrochemical gradient. So it could be of two types, either the simple diffusion or the facilitated diffusion. This simple diffusion is unassisted. It means it does not require any kind of help. It will be carried out from higher concentration to lower concentration. है ना? So it occurs normally to the phospholipid bilayer. Whereas this facilitated, as the name itself suggests, this movement will be facilitated or you can say assisted. So it occurs to the transmembrane. So, this facilitated diffusion can be saturated at higher concentration of solute. Both of them are just movement taking from higher to lower only, but this facilitated is assisted, but this simple diffusion is not assisted. So you can just see here a structure of plasma membrane that is a phospholipid bilayer. Along with that, the protein immersed as well as the carbohydrate. So some of the ions or many other substances that are not allowed to pass, you can just see they will be reflected back or will come back. Then some of the large molecule, although they cannot diffuse easily, but they can pass through the assistance. They require a carrier mechanism or the carrier protein. I will let you know about the carrier protein. Then some small water soluble molecule and ions, they can easily diffuse. So these membrane channel. You can just see a kind of a flow chart. है ना? it could be transport could be of two types broadly, passive as well as the active. So passive, simple diffusion, osmosis as well as the facilitated diffusion. Osmosis is a, you can say, a example of diffusion only. Only the major difference between the diffusion and osmosis is that in osmosis we talk of the movement of only and only solvent, whereas in case of diffusion we talk of the movement of the solute as well as the solvent, means solid, liquid, gas. Both of these are considered in diffusion, whereas only the liquid movement is considered in osmosis. Then in case of active, it could be either be pump or the vesicle. I will let you know. Pump further can be of two types. Active transport, you can just see here, it is against the concentration gradient. So primary active, secondary active, uh, ATP driven, primary active. So unique ions are transported through such mechanism, like that of the sodium potassium pump, calcium pump in muscles, and a proton pump in case of stomach. Whereas this couple transport, they use a downhill transport energy, है ना? It could either be of two types, import and antiport. When two things are just transported in same direction, that is the symport. If in opposite direction, that is the antiport. So in the example, glucose sodium pump, sodium calcium exchanger, sodium phosphate co-transporter are a well-known example of the coupled transporter or the secondary active transport. Then the third one more is there, light or redox reaction, bacteriorhodopsin. In case of prokaryote, we observe such bacteriorhodopsin is present in bacteria only, and such transport is being observed in them. So you can just see here uh, these lines mentioned in pink color. The absorption of the products of carbohydrate and protein uh, while digestion are against the concentration gradient, whereas the lipids are absorbed by diffusion. So very, very important. That's why mentioned in different color. So this carbohydrate and protein can easily be digested. Means they just transported against the concentration gradient, whereas the lipids are absorbed by the diffusion. Here you can just see a difference between the simple diffusion and the facilitated diffusion. In diffusion, I just have told you now two types, facilitated and simple. So here you can uh, easily will be able to differentiate. In case of a simple diffusion, we talk of the transportation of the non-polar particles, whereas in facilitated diffusion that is assisted, we talk of the transportation of the polar and the large particle. This simple diffusion occurs directly through the cell membrane, and this facilitated diffusion occurs through specific facilitator molecules. Then rate of diffusion is directly proportional to concentration gradient as well as the membrane permeability of the solute molecule. Whereas in case of facilitated diffusion, it depends on the kinetics of the carrier mediated transport. The example is mentioned diffusion of gases, diffusion of molecules from blood to cell through the interstitial fluid. Whereas in case of facilitated diffusion, chloride, bicarbonate, transport, counter transport in case of the excretion, है ना? Co-transport of sodium with sugars like glucose, galactose, etc. are the example of facilitated diffusion. Then yes, if we are saying that some assisted molecules are there, what could be they in case of facilitated diffusion? They could either be carrier protein or permease or the channel, है ना? So these carrier or the permease undergo conformational change to transfer the solute. This is a slow process, and this carrier protein is involved in passive as well as the active. In passive, in facilitated diffusion, these are involved. Then in channel, they are involved in the uh, passive transport and form continuous pore across lipid bilayer. These are faster. Then three protein are there. Flippase, floppase, scramblase that are involved. Flippase will flip, floppase will scramblase in both of the direction. So, lipid bilayer is maintained due to slow diffusion, protein-lipid interaction, as well as the protein-mediated transports. If we talk of the motion of the lipid molecule, it is not rigid and static state structure. है ना? Rotational movement is observed, lateral movement is observed, as well as the flip-flop movement is being observed. Fluidity is, depends on the chain length as well as the unsaturation. If lipid is it with short chain and unsaturated fatty is still they will undergo a transition. है ना? So at low temperature, you can just see at low temperature what will be the situation or the scenario of the plasma membrane, whereas under a high temperature, the fluidity will increase, unsaturation will increase. Cholesterol, then at high temperature, fluidity decreases. At low temperature, cholesterol prevents membrane from the freezing. So that's all for today. Thank you.